with Dr. Marynka Ulaszewska.
Thank you, Claudio. Thank you very much. Let me share my screen.
Yes.
And let me do the presentation mode. Can you see that?
Yes.
Okay, and I hope you hear me well.
Now is in the presentation mode.
Okay, lovely.
Yes.
So thank you.
Thank you.
May I start?
You may start.
Okay.
The stage is yours.
Okay, thank you, Claudia. Okay, so thank you so much for the invitation and hosting the topic about the food and the nutrition and the mass spectrometry. My presentation will be dedicated to the nutrition and untargeted metabolomics, basically the analysis of the urine, plasma, and serum. You must know that the Nutri-metabolomics is an excellent platform to study the interactions between the human metabolism and the food intake. You can measure and monitor the consumption and the absorption of the nutrients within the several time frames, like minutes or hours. You can also follow them within the years or even entire lifespan, checking these interactions and the impact of the food intake, the dietary patterns. The Nutri-metabolomics is excellent to study the primary and the secondary metabolites, check and investigate the improvements of biological functions, of course, the elucidating the metabolomic pathways, underlying the different diseases and the phenotypes, and to elucidating the dynamic responses of the system metabolism into specific challenges, which in the health and the disease state, and in this case, that would be the food intake. This is the general untargeted metabolomic workflow that was developed, and it basically works like this for not only for the Nutri-metabolomics, but for any metabolomics experiment. So that's of course starts with the research question, with the study design, with the generation of the data, with the collection of the metadata, and then the conversion of the files, the processing that consists of picking, grouping, the quality check, the imputation of the meeting values, then the explorative analysis to verify any batch effects or outliers or intensity loss. And then you go through the data analysis and statistical modeling, then of course the annotation, which is probably the bottleneck today, and then the results and the validation of your data. If you would like to know a little bit more about how to design and how to execute the Nutri-metabolomics experiments, I'm warmly inviting you to see this paper about the integrative action for the nutritional metabolomics in the football projects. That was the fantastic joint partnership initiative, and me in the past as a member of the Foundation Animals Mark has this pleasure to join this project and to write this paper, which is a kind of a compendium of very practical knowledge and the lab know how many tips and tricks how to do and what to do in the lab, and also what to do with your data. So those few examples on how to design and what kind of design studies you can find, how to collect the biological fluids, several sections, I think 11 chapters in this book. You can find a lot about the annotations, a lot about the quality control, about the randomization, about the normalization of the samples, really many of information. So I'm really inviting you to have a look on that. But I would like to show you some practical examples on what to do and how is the Nutri-metabolomics, so the study of the food and the human health interactions. And this is the first example. So that's the workflow that was developed in the Foundation Edmund Park some time ago. It works really greatly. I will show you a few examples. That's an example of the urine that was extracted on the 96 well plates, separated on the C18 column, analyzed with an Orbitrap LTQ XL machine on untargeted fashion, with the high resolution in the full scan mode in an MS mode. Then we applied the statistical analysis on it, and then accomplished the study with the biomarker identification and the structural elucidation. The part of the this is structural elucidation is the one that requires the most effort today, and the analysis usually consists of deep verification of the full scan information, checking the shape of the total ion chromatograms and extracted ion chromatograms, as you can see on the left. You can find in the full scan not only the precursor ions, but also some species that belongs to them, like loss of water or loss of the glucanorite moiety. And then you squeeze as much as possible, and you assign that the elemental composition to the PQC, and you see that it's statistically significant. And in the end, you perform a mass analysis. You compare them with the library, which is free available. It's an MSIT cloud, very nice, with a great algorithms to look for the substructures matching between your sample and the standards that are available in the library. That's an example on how we did the annotation of the Abyssic acid glucanorite moiety within MSIT cloud. So the examples. The first example will be the short-term study, multi-compartment and multi-Amix analysis of the intake of the apple juice. We had the 12, that's the design, the study design reported here. So the 12 healthy volunteers drink the 250 milliliters of the cloudy juice, or on the other hand, the enriched with the apple polyphenols cloudy juice. After the analysis, we found really many compounds that moved within the postprandial hours when we collected the plasma and the urine samples. So you can see here the examples of metabolites that were increasing within the time in the biological fluids, like for example, the fluorescein glucanorite with the Tmax at 1 hour postprandial in plasma, and or on the other hand, compounds that never reached the maximum of the concentration, either the Tmax, like for example, the hippuric acid, which is on the right side corner, down corner. You may appreciate here really many different shapes of the kinetic curves for each metabolite. The dashed line is the one corresponding to the normal cloudy juice, and the black normal line is corresponding to the enriched apple juice. So you see here also the certain dose response relation. What's important is to understand where those metabolites comes from. And that's the example. I hope that you can see it correctly and with a good resolution. In the green color, I reported the names of apple constituents, apple ingredients. And in the violet color, I reported metabolites that we found to be statistically significant and circulating in the biological fluids, and the link between them. So where each metabolite come from. So we see that the procyanidins get through the flavantriols like catechin and epicatechin give a rise to the many hydroxyphenylvalerolactones and hydroxyphenylvaleric acids with a different hydroxylation groups on the phenyl ring. On the other hand, the hydroxyalcones like fluorine and fluorescein, they give rise to the phase 2 metabolism. So the fluorescein sulfate or fluorescein glucanorite, like on the left side. You see that in these blue boxes, I was trying to keep together the compounds that were structurally similar, but there are many of them that are in the middle out of any box. And those are the ones where the impact of the microbiota is very important, and the interaction between the host, so the host metabolism and the human metabolism, the microbiota and the food, they all together play an important role in their production. When we think about the correlation of metabolites with microbiota species. That's also super interesting topic, as many of those metabolites are produced thanks to some species of the microbiota bacteria, let's say. And that's the picture showing you the correlation. You can appreciate here in the green box down very similar patterns in the responses between the correlations between the metabolites and the microbiota species. So you see that they are very characteristic. It's not completely like a random situation when those metabolites, by the way, those metabolites belong to the same group. They are structurally very similar between each other. So the core of them is the ferulic acid. That's very interesting. The second example I would like to mention today is the long-term effect of the apple consumption on the human health. And that was one of the projects that was executed in the collaboration between the University of Reading and the Fondazione Edmond Machin Trento. And I also was very happy to give my input here. It was the long-term study, as I said, with the apples. That was the Renetta Canada variety versus the consumption of the apple juice, which was the very much aromatized water. That's the study design, so quite long. 8 weeks altogether. It's 22 weeks of the trial with the particular monitored inclusion criteria. The objective here was the mildly hypercholesterolemic patients. Several biological fluids were collected here. So the serum, the urine, the fecal samples. Many clinical measurements were also taken here. But we are talking about the metabolomics. So let me show you a few examples of the results from the metabolome profiling. And that's one of those. So we have seen the statistically significant reduction in the total cholesterol and LDL cholesterol. That was a good, very good outcome. We also saw several relations between the total cholesterol and the bile acids, and the differences also between the males and the females. But when we talk about the untargeted assay, I would like to show you those few graphs showing you the metabolites that were found statistically significant. Some of them were belonging to the tryptophan pathway, like here marked in the blue color. So the tryptophan, the indole, acryloid glycine, and the can you see this one?
Yes.
I will go very fast. This is the third example with a dose response study, the dietary intervention with low and high flavonoids, fruit and vegetables given to the free living population with a high risk of cardiovascular diseases, very particular exclusion and inclusion eligibility criteria. I will give you the link to the paper so you can read a little bit more about that. This is the study design with the increasing doses of the fruits, of the fruit and vegetables within the several 18 weeks of time. Red vegetables versus green vegetables versus the habitual diet. It's a UK diet, right? So it's a long time, it's a free living population. So the participants received the fruit and vegetables at home, and they were encouraged to follow the diet with 24-hour recalls made by trained nutritionists. Those are examples of these red and green vegetables that were included in each of the dietary interventions. I will go first directly to do not lose much of time. I will go to show you the compliance with the dietary interventions. So are we sure that the people eat what they were supposed to eat? Yes, they did. So we have here the total flavonoids in the fruit and vegetables versus the total flavonoids in the urine, and the trajectories of each of groups is corresponding. The same for the vitamin C intake. So there's the total vitamin C content in the fruits and vegetables and the plasma vitamin C concentrations. And also here we see the high compliance with the diet. And in the end, the carotenoids were designed to be the same in the two dietary interventions versus the control group. Or also here there is a coincidence. So many very good outcomes after the dietary interventions, especially in the men. So the increased endothelium-dependent microvascular activity, and there was a reduced C-creative protein, and many more. But what about the biomarkers of the food intake in the people here in the urine? So one of the compounds I found to be statistically insignificant was the Abyssic acid glucanorite. That's an interesting compound. This is a plant hormone, and it's an intermediate step within the plant in the synthesis of carotenoids. But also the Abyssic acid can be released from the glucose, challenge the human pancreatic cells, and stimulates the insulin secretion. So there are two hypotheses for this compound. One was the carotenoid metabolism, and the other one, the increased level because of the human pancreatic cells. But as I said before, the carotenoid level in the dietary interventions, they were the same. They was designed to be the same. So the second hypothesis was the more convincing. How did I find this compound among many features that were to be analyzed? So first of all is the analysis of the precursor ions and analysis of the fragments. Here I was able to find not only thanks to the data-dependent analysis made with an Orbitrap, I was not only able to find the MS2, but also the pseudo-MS3 spectra, so the MS of some neutral losses in the full scan. Then the comparison with an MZ cloud and the purchasing of the analytical standards. But here, please be very careful, because the Abyssic acid is very similar in the fragmentation pattern to the carboxyethyl hydroxychroman, which is one of the vitamins. That's the derivative of one of the vitamins. And you see that there are at the level MS2, at the MS3, and even at the MS4, there are similarities in the fragmentation patterns. The main differences are due to the ion intensities. So please be very careful when you do that. The other metabolites were related to the hippuric acid, and it was a cyclohexadiene carboxylic acid glycine and cyclohexadiene carboxylic acid glycine. So the names are almost the same, but you see the structure. There are two double bonds more in the cyclohexadiene, while there are no double bonds in the cyclohexane. They both belong to the hippuric acid pathway. But I said, okay, so if I have two compounds that belong to that pathway, maybe I have something more. And I was analyzing the data, looking at the trajectories, so looking at the excretion of these metabolites within the time frame. And if you can see the green trajectories in all four graphs, they're very similar, showing that indeed they were produced probably within the same metabolic pathway. And indeed, all of them, two of them were statistically significant, while for the older compound, the p-value was 0.08, and the hippuric acid was not statistically significant. But this is mainly due to the high inter-individual variability of the person that was participating in the study. And again, it's important to understand when these compounds, where they come from. So if you have to do something with the compounds that have a different level of the saturation and benzyl ring, you need to think about what's the structure that allow me to obtain those compounds. And if I deal with the fully aromatic substrates, it's very hard that I receive after the metabolism differently saturated or not saturated intermediate metabolites. But on the other hand, if I have at least one saturated heterocycle in the molecule, I may be able to receive, to find after the human metabolism, differently saturated intermediates. And these compounds, which I'm indicating now, is the chlorogenic acid, the one that was actually very popular and highly abundant in the green diet. This is what happened with the compound in our body. So once the chlorogenic acid enters the body in the small intestine, is divided into two moieties, which one is the hydroxycinnamic acid. You see 3 double bonds and benzene ring, and the chinic acid, so the full saturation. And from the chinic acid, we can start our journey to produce the cyclohexane, and cyclohexadiene carboxylic acids. Those give a rise to the benzoic acid, and both, actually all 3 compounds and also benzoic acid are able to get from the intestine, through the intestinal wall into the liver. And in the liver, they are conjugated to the glycine. And this is the compounds we found in our study. There were many more. There were so many metabolites being statistically significant through the different time points in this study. One of them was the proline betaine as the marker of the high flavonoid diet. And if we think about the origin of the proline betaine, we know that it comes from citruses. And indeed, the citruses were allowed in the high flavonoid diet. And this is on the plot right hand down. You can see the intake of the oranges and the grapefruits. And it's a kind of similar, maybe a little bit arbitrary, but kind of similar to the trajectory of the proline betaine expression. The other one was the iberin and acetylcysteine. That one was really hard to be annotated. I must be clear in this point. But also this one actually reports very nicely the dose-response correlation between the intake of the radish and the red cabbage and the trajectory of this red line here. Many more also for the red diet. Many fatty acids moving upregulated there. And finally, one of the last one, I would like to put your attention on the importance on the fragmentation parts similarities. This is the biomarker found after the berry intakes. That's a tetrahydromethyl carbolene decarboxylic acid. The structure is depicted on the left side down. That was the easy one because we had the analytical standards to be compared, but I found also another compound that was behaving very similarly, and the fragmentation pattern was also similar. Also, the numbers are not really the same, but you must know that the point here was to figure out the delta of 4 daltons, the differences of 4 daltons between the precursor ions, but also between all the fragments. And that was the main point where I found out that those compounds are correlated, and the only difference was the presence of the 2 double bonds. If you would like to know a little bit more about the human metabolism and the food intakes and the correlations between them, I'm warmly inviting you to see the website of the Joint Partnership Initiative project, which was the FoodBal, the Food Biomarker Alliance. Many exciting papers about this topic. Different techniques were employed to study the responses in the humans. There was the NMR, there was a GC, and there was a high resolution LCMS. You could appreciate the biomarkers of only one intake of particular food, but there were also amazing kinetic curves that were similar to all intakes, showing that whatever you eat, you may have the similar response to that. And with this, I would like to thank you for your attention. I hope I didn't exceed too much time. I hope so. Thank you.
Thank you, Marynka, for your very interesting presentation. So, any questions?
It seems there are no.
No question. I have a question about microbiota metabolites.
Yes.
Yeah. Do you think that it's their presence is also recorded in blood or in...
Yes, absolutely, yes. So we found within this kinetic studies that indeed after certain times, so usually it's like maybe 4, 5 hours, 6 hours, and later on, the metabolites that come from interaction between the host and the microbiota, they start circulating also in the plasma. Yes, absolutely, yes.
It's common to absorb them from...
Yes,
from
they are absorbed from the intestine. Yes, of course, and then they are circulating. With their presence and their concentrations are not the same in the urine and in the plasma, of course. In plasma, usually you see the concentrations are significantly lower, like not everything that we eat and we produce circulates, otherwise we would be, that would be a little bit dangerous. But with a certain, to some limits, yes, their presence is there.
Okay, thank you very much.
Thank you once again. I'm sorry for this interruptions. I was, emotional. Thank you so much.
The problem of our times, of our online life.
No, this is the problem of the countryside that my mom is living. Thank you once again.